Hi there, welcome to the DNA lecture video number 2. This section of the lecture notes, we're going to be talking about RNA, and we're going to be talking about bonding, base pairing rules, and complementary base pairing rules. So let's begin. The three types of RNA that you need to be familiar with are called mRNA, messenger, transfer RNA, and ribosomal RNA. Let's first start with rRNA because it's real simple. Ribosomes are the protein factories inside of our cells. They are not located in the nucleus. This would be the ribosome over here. This is nucleus, ribosome out here. So this ribosome is made out of proteins and rRNA. So ribosomes are made out of ribosomal RNA along with proteins. There they are, outside the nucleus. So that's the rRNA. mRNA. This is the messenger RNA. Well, what does that mean? Well, DNA is a code stuck inside of the nucleus. This code needs to get out of the nucleus and to the ribosome in order to produce a protein, because this whole this whole unit is eventually about producing proteins. So mRNA is the messenger. It is a small code compared to the DNA, and it is a copy of a gene for the most part. All right, so messenger RNA takes the genetic code from the nucleus to the ribosome. That's the job of messenger RNA. And it is a copy, a small copy of the DNA. And it is usually a copy of the gene that makes its way to the actual ribosome in order to produce a protein. tRNA or transfer RNA, they transfer amino acids. Well, what does that mean? Well, these little circular dots here, these are amino acids, and they're floating around inside of your cell. This is what we get them from our food, from our nutrition. So you eat protein in your food, your body breaks it down, turns it into amino acids. Now you have amino acids floating around ready to build protein inside of your cells. Well, transfer RNA or tRNAs, these guys right here, they carry those amino acids to the ribosome in order to build the protein. So their job is to bring over the building blocks of protein, the amino acids. Okay, so mRNA, the message gets from the nucleus to the ribosome, right? tRNA carries the amino acids to the ribosome to build the protein, and RNA, that is what the ribosome is structurally made out of, right? Um, let's begin. Next one. And by the way, you're going to see this word transcription come up. We're going to have a whole lecture on it. It's not quite yet, but transcription, all that means is copying. It's rewriting or copying the DNA. So when the DNA gets transcribed or goes through transcription, it gets turned into one of these types of RNA. So how is RNA made? It is made by this process called transcription. We will have a whole lecture on it coming up. You have not quite seen it yet. So all RNA is made by the process of transcription. Structurally, RNA is very similar to DNA. Okay, nucleotides is what RNA is made out of. All right, so RNA and DNA are both made out of nucleotides. It is the building block of DNA and RNA, but there's structural differences. What are the two differences we're going to see? Difference number one, DNA has a sugar called deoxyribose. RNA has ribose. That's what the R stands for, ribose. The D in DNA stands for deoxyribose. So they both have 5 carbon sugars, but they are different. Okay. They both have phosphates. That is no different. They both have the exact same type of phosphate, no change in the phosphate. Both RNA and DNA also have nitrogen bases, or we call them nitrogen carrying bases. Same thing, right? There's a difference though. RNA has a base called uracil, U. DNA has T's instead of U's. So you will never see a U in DNA. So if you ever see a U, you know for a fact it is not DNA. If you see a T, then you know that you have DNA and you do not have RNA. So it is a structural difference. The U's replace the T's. Okay. And this just showing you a picture right here. If you look at the picture, they're very, very, very similar. You see this oxygen here and this oxygen, one less oxygen. You have one extra oxygen here, one less oxygen here. Deoxy. That's that one deoxy, one less oxygen. That's the difference between the sugar. So ribose and deoxyribose has one less oxygen. Okay. The bases, this is DNA. How do we know? It has a T. We have a U here. That's how we know this one's an RNA. So they call them RNA nucleotides, DNA nucleotides. RNA, it's small. It's usually short. DNA is very long. That's a structural difference. DNA is really big. It's so big it cannot leave the nucleus. RNA is small. It can leave the nucleus. RNA is one strand. DNA is two strands. So RNA is a single-stranded molecule. DNA is a double-stranded molecule. RNA is usually a linear shape. DNA twists up into a ladder. Okay, so RNA can be linear. DNA is a double helix. RNA is single. DNA is double. RNA is usually small or short. DNA is usually really long. DNA has T's. RNA has U's. RNA has ribose as the sugar. DNA has deoxyribose as the sugar. So there's structural differences. Okay. The two types of bases in DNA and RNA, they're classified as either a purine or a pyrimidine. Purines or pyrimidines. All right. The big difference is this: purines are larger in size. Well, what makes them larger in size? They have a double ring, which makes them longer. Okay, these are rings of carbons. They have a double rings. Pyrimidines have a single ring, a single carbon ring, so they are smaller. When they pair together, a larger one like A always pairs with a smaller one like T. A and T always bond together because they produce the correct number of hydrogen bonds. G always pairs up with C because they produce the correct numbers of hydrogen bonds. I'm going to show you a picture in a second. So A's always bond to T's, G's always bond to C's. A purine can only bond to a pyrimidine. A can never connect to C, and if it does, that means an error occurred. If G connects to T, an error will have occurred. A's and G's cannot connect to each other, and C's and T's can never connect to each other. All right. So you always have a purine connecting to a pyrimidine, specifically an A to a T and a G to a C. Uracil is not shown here, but uracil is a pyrimidine and it replaces the T. So uracil is a smaller one in size, like thymine. All right. So it shows here thymine is a pyrimidine, uracil is a pyrimidine. But uracil is an RNA and thymine is in DNA. Okay. I mentioned to you a second ago, purines and pyrimidines always bond together and in a specific fashion. A or adenine always binds the T or thymine, always. Two hydrogen bonds are made, and that is how and why they know to attach to each other. C's and G's and C's produce three hydrogen bonds, as shown to you here in the picture. And that is why G's and C's always bond to each other. So in this case, the A and G's, these are the purines. They're binding to the pyrimidines over here on the right. Okay. And they always bond according to the number of hydrogen bonds that they can make. 2 hydrogen bonds for A's and T's, 3 hydrogen bonds for G's and C's. And these are weak bonds, basically, not covalent. Covalent are very strong. So we have what's called complementary base pairing rules. So here's the deal. DNA, and you'll notice start isn't at the beginning, and that is because sometimes DNA is used to make DNA. Okay, all right, that goes from the left, from DNA here to the left. Sometimes DNA is used to make messenger RNA. So you would start here and you go there. So you would not jump all the way here. So the start point is at the second spot, not the first spot, because it depends on what you are producing. So let me give you an example. DNA to DNA. Okay, so we would start here. A G would go to a C. G would go to a C. So you would write down a C. The next letter. The next letter. A C becomes a G. So right now here you would write a C. Then we have a G. Then we have an A because T corresponds to A. A corresponds to T. T corresponds to A. C to G. G to C. A to T. And T to A. All right. I'm going to give you all of these in a second if you did not get a chance to write it down. Okay. Better yet, let me show it to you right now. All right. So these are the corresponding or complementary bases that go with DNA to DNA. From DNA to DNA, this is called DNA replication. You're producing more DNA. This is a process called DNA replication. All right. So we start here. We went to the left. All right. Now I'm going to explain to you why we start here and not at the beginning. Well, you start at the second point because sometimes you need DNA and you're going to rewrite it or copy it as mRNA. This is called transcription. And like I said, we'll have a whole lecture on it later. Transcription is taking DNA and converting it to mRNA. All right. So you don't have to go from DNA to mRNA. No. Sometimes you just go from DNA straight to mRNA. So in this case, T's become A's, A's become U's, G's become C's, and C's become G's. So look here, C to G, A to U, T to A, G to C, A to U, C to G, T to A, T to A, and G to C. Okay, so these are the complementary bases that are mRNA bases that are complementary to the DNA. There's no T's here. On this first one, these are the complementary bases of DNA that go with the other DNA. So the DNA is complementary DNA. This process would be called DNA replication. This process here would be called transcription, turning DNA into RNA in this case. All right. Finally, number 3, sometimes you go from mRNA to tRNA. Well, when does that happen? It happens in this process called translation, when you're producing a protein. A's go to U, U go to A, C to G, and G to C. Okay. If you notice in the RNA columns, there's no T's. Okay. So U's have now replaced the T's. So here we go. U becomes A, G becomes C, G becomes C, C becomes G, A becomes U, A becomes U, U becomes A, G becomes C, and G becomes C. So once again, these are the complementary tRNA bases that go with the mRNA bases. These down here, complementary mRNA bases that go with the DNA. And over here, complementary DNA that goes with the original DNA. All right? All right, we will stop there.